We should make a start because it's just about 3 o'clock now. So this is the first in what I hope is going to be a successful series of research showcases where every week, in this semester and next semester, a PhD student or a RA, research assistant, will come and tell you about interesting research stuff. And this week it's Dave Thorne from the Advanced Interfaces group. Dave was an undergraduate here starting when? Do you want to divulge that?
99.
99, and did a 3-year course.
Yep.
And got a bachelor's degree. And then stayed on, did PhD.
Yep.
Did PhD with Steve Pettifer in the Advanced Interfaces group. And then is now a full-time researcher doing Utopia documents and other things. So, welcome Dave. And
thank you, Toby. So yes, thank you for coming. Good afternoon. Welcome. As Toby just said, I'm going to tell you a bit about my research over the last few years, but I think to begin with, it may be interesting, it may be not, but I think I'd like to tell you a little bit about how I see research and why I think people should do research. There are worse reasons to do research, to go into doing a PhD, for example, than convenience or because you want to have a doctor at the beginning of your name. But I do think there are far better reasons. Research to me is about investigating the bounds of human knowledge. And that sounds very, very lofty. But it is what research is about. It's about seeing, for a particular field, how far into it you can go, find what people in the world know about it, and ultimately to push those boundaries back. And that, I think, is a fantastic reason to do research. And if you keep that in your mind, you should at least do good work. Perhaps not profound work, but certainly good work. Now, having said that, PhDs, by the time you finish a PhD study, you should be the world's expert in one particular field. Now, that does not mean computer science. The field of study is a tiny sliver of computer science. And it might be, I should definitely say, probably no one here will do anything as profound as invent the internet. And I know I certainly didn't. On the other hand, for a given little area, you might try to solve a particular problem in a new way, thereby getting insights into that problem and how we might be able to solve others. And slowly, but surely, that boundary gets pushed back by all the little advances. And occasionally, you may be lucky enough to be able to take those advances and do something very, very big, very good. Like cure cancer, for example, though in computer science we're unlikely to do that. We could well build the computer system that would allow someone else to cure cancer. And I personally think that's a fantastic reason to do research. Certainly the reason I do it. With that in mind, my history of research has basically been trying to work out how to allow scientists to be more efficient with what they do. A number of years back, probably by now 8 years ago, I started doing research into how biologists use computers, how visualization and analysis on computers are done in aid of biology, and also, more importantly, how badly it was being done in various fields, and how we can fix that. So, one of the very first things I did was look at how to use novel graphics techniques in how to render very, very large molecules on the screen, interactively, such that a biologist can look at it in 3D, move it around, investigate it in a way that before would have been clunky at best. Because if you wanted to use, say, ray tracing techniques, ray tracing can take a long time, which means just turning a molecule is like watching a slideshow. Whereas employing proper computer science techniques for this kind of thing allows you to move something like that around absolutely interactively. Moving from there, we then, as a bit of a subgroup, developed within the AIG. We looked at how various different kinds of biological analysis can be done on computers. However, better than they were before. This is a piece of software called Utopia. It's not Utopia Documents, which I'll introduce in a moment. It is the old Utopia. But essentially, it allows a biologist to take very complicated data about protein sequences, about DNA sequences, and about protein structures, to be able to link them all together, work out where on structure certain things are in the sequence. If anyone here knows anything about biology, that will hopefully make a bit of sense. But it also allows you to do analysis that in previous years would have taken a very long time. So the old way of doing things in biology would be, you take, for example, a sequence of letters which represents a protein, and you might do a bit of copying and pasting, putting that into a website. That might then go away and do some analysis and come back, maybe even email you the results back, which you then copy and paste back into probably Microsoft Word, where you highlight some things, copy it back out, put it in a program, by which point, 30 minutes later, you've probably miscopied half the stuff that's gone through here. And in the end, you get something which not only has taken a very long time, but you're not entirely sure that you've done the right thing. And it requires from the biologist so much complicated computer science knowledge that it meant their job was hampered. So what we did was we built a piece of software that tried to hide all of that underlying complexity. So that in the end, if they selected a couple of things, right-clicked, and then you provided information to them in language that they understood. They press a button, and it goes off and does all of the complicated stuff behind the scenes, comes back, and from their point of view, they've just done what they wanted to. And it gives their brain more space to think about the problem they are actually trying to solve. Because it's our problem to solve sending data to servers and wrapping things in XML. That's our problem. It's not supposed to be a biologist's problem. So Utopia did that. And then we slowly went from biological data to biological literature. So I have no idea of the makeup of this audience. I don't know how many people here are undergrads or postgrads. However, if anyone here does go into research or is already in research, you will know what scientific literature is. Apart from a headache, it tends to be maybe an 8 to 12 page document, very dense in text. It might have a couple of graphs, it might have some images, it might have some tables in it, but that is the workhorse of scientific research. Certainly in biology, but also in chemistry, physics, computer science even. Although we tend to use our literature differently in computer science, in biology, most of the biological knowledge of the human race is locked in its literature. And that is an important thing, because at the moment, it is locked there. Someone once said that a scientific article is a story that tries to tries to convince you of a particular subject or a particular idea, a particular thesis, but it has to have data behind it. And those two things, the story and the data, and the reason they don't really fit together, that makes computer science access to literature very, very difficult. So there are various problems. Natural language. Pretty much every single publication ever done is in natural language. Possibly not English, obviously it could be any language, but it's certainly natural language. People don't produce publications in RDF XML that can be reasoned by a computer, because no one would be able to write them. And so they're written in English, which that's the first hurdle. Getting a computer to understand English is itself a complicated task. If it's simple language and unequivocal statements, we're quite good at that. However, with scientific publications, that is never the case. You never find simple language, certainly in biology, you never find it in scientific literature. People try to make complicated sentences. Not only that, they try to couch everything they're saying in as much caution as possible. Because if they try to publish a thing saying, A does B, they'll probably never get published. Because it's too bold a statement, it's not necessarily something that you can say according to the evidence. And so that statement will end up becoming, A seems to, under these circumstances, with these levels of assurance, might do B. Now, getting a computer to understand that that meant that they think A does B is pretty much impossible. So natural language and cautious language is a bit of a bugbear, it's a bit of a bind for us, people trying to get at the data. Also, it's unstructured. People have different, inconsistent ways of saying things, of naming genes, proteins, even IDs into databases. Some people might have prefixes on them, other people might not. There are many things that if you weren't a biologist, for example, and you didn't know exactly what the paper was about, you'd have no idea what the majority of the words meant. And if a human who has a lot of context and knowledge can be confused by that, then obviously a computer can. PDFs, that's another unfortunate problem. The PDF, the portable document format, it's a fantastic format for showing how something can be printed, how it should be displayed on paper. But it's not very good for analysis. Essentially, it's a big bag of words, and those words have got coordinates, so it knows how to reconstruct the visual page. But a computer really doesn't like that. A computer wants to know that this bit is a heading, that this bit follows on from this bit, that these two columns are actually two different columns. PDFs don't have any of that information. So getting access to information from PDFs is difficult. And finally, the other side of the persuading of data is the data itself. If you read a scientific article, you might be lucky to have maybe a couple of graphs, maybe a couple of tables, a couple of images. The chances are some of those things will only have been put in to kind of zhuzh it up a bit. Really, so it's not just blocks of text. The data behind the experiments that a given piece of literature is talking about won't be in the paper. This might be because you can't have 400 million pages because there's 2.5 terabytes of sequence data behind what you're reading. And sometimes it's because you're not allowed to see it because it's a pharmaceutical company's data and they don't release them. Therefore, getting access to data that's behind a given article is, if you're unlucky, impossible. If you're lucky, it is possible. However, if it is possible, it's probably not easy to get to. There aren't very many tools that can just take you from one PDF to the data it's talking about, because nobody enforces rules to allow that to happen. So different publishers might have different ways of doing it. Most of them don't do anything. So they're the things that you might not like about the scientific literature, and certainly the things that annoy me. So now I'll explain Utopia documents in a moment. It's a piece of software that we've designed to try to solve all those problems. But just to put something in context, I've now been talking for about 15 minutes. Is that right? So Pubmed is a database of scientific articles in the biological and medical realm. It's a very large database. It has grown exponentially over the last few decades. And as of the last count, which was the last count I did of a couple of years ago, it had one new scientific article submitted to it every 30 seconds. So, since I started talking, there's been something like 30 articles. This is about 30 articles because I measured how long I was talking. About that much information has been published since I started talking. That's all just in biology. We're not talking about computer science, chemistry, geology, oceanography, nothing like that, just biology and medicine. There's no way any one person could read through that amount of information in 15 minutes. Granted, quite a lot of that stuff will probably be irrelevant, but you need to read a certain amount of it to be able to know it's irrelevant. So when it gets down to actually being able to read the stuff you care about, efficiency is key. Just have to put those on. 30 seconds. I won't do that for the rest of it because I'll forget. However, so I'll give you now a quick demonstration of Utopia documents. Kind of the Simplest thing you could say about it is it's a PDF reader. It'll open a PDF and let you view it. However, if what you've opened is an article, in this case I'll open a scientific article. It's a biological article, I think. Something to do with drugs, antidepressants and cell membranes. Ah, is it too light? Is that better? So this is an article. I need to quickly log in to our wonderful Wi-Fi. Bear with me. That's better. Start again. There we go. Good. So what normally would happen if you were to use, say, Adobe Acrobat or Preview on a Mac, if you open up a PDF, you just get a PDF, you get nothing else. If you want to then do your own investigation, you've got to do it yourself. Utopia Documents is a bit more clever than that. It does various things. It fingerprints the document, it looks at the content, it tries to work out what the title is, who the authors are, the general idea of what the document is about. It then throws that to various servers and comes back with as much information as it can, verifies that, and displays it in this sidebar on the right. You'll notice it's worked out the title, obviously, but it also gives you direct links to various identifiers for this document, including links to the publisher. Gives you an ability to change and download various citation formats in case you wanted to include that in your own research. I'll scroll down a bit further, gives you altmetric information, and this is, when it's been mentioned on blogs, or it's been posted to Google+, or someone has read it in Mendeley, all those things go together to tell you how important the paper is that you're reading. If you open up a paper and it looks quite quickly as though this paper is nonsense, maybe it's already been refuted, or even it's been retracted, that information will appear immediately as well, allowing you to discount papers quickly if it turns out that you really shouldn't be spending your time reading it. Just various other things which, as I just mentioned a minute ago, take a very long time otherwise. So Figshare is a service, it's not a service that we've done, but we have included it, which allows you, given any particular article, to see whether or not anyone has posted the auxiliary information for that article. And that means the data. That data I said a minute ago that's almost impossible to get in most cases. If it has been provided as a Figshare article, then you can get to it straight from here. And this article itself has got quite a lot of stuff. I won't click through because we'll be here forever. But it has a lot of auxiliary supplementary materials. It gives you even information about what you're allowed to do with the article, because one thing you'll learn eventually is that you're not necessarily allowed to do some things with some articles, depending on what their publishers said you can. Most publishers, if you look at their kind of terms and conditions, almost prohibit you from reading it. They're so strict. I mean, obviously you have to be able to read them, so you can pretty much ignore that. But you have to be careful what you can do and what you can't. And another thing which is, Google Scholar is very good now. It means that you can, given an article's title, try to find very quickly where that article is online. But it still takes quite some time. It also takes you, if you're reading this article, for example, if I scroll down, this sentence here, I don't know whether or not that highlight is visible. That sentence there, at the end of it, there is a citation, it's number 4. Normally when you're reading a document, you come across that. You need to probably go to the back of the document, look in the bibliography, try to find that document, get enough information from that document to then maybe go to Google Scholar. If it can find it, if it can't, you might have to search Crossref or Pubmed. Once you've done that, you might be able to go to their website. If you have access to it, you can download it probably. Otherwise, you might only be able to see abstract information. However, because Utopia Documents is clever about things, it knows where the bibliography was held by the publisher of this article. It knows how to then use that to go off and find other information behind the scenes, such that just clicking on that particular number 4 in brackets now gives you a pop-up that lets you try to find more information. And if it's already found the PDF, there'll be a PDF, for example, which means that what was just a moment ago a 10-step process that takes possibly 5 minutes for every single bibliography entry. Now, you just download it, press a button, all gets done in the background, and you've got it open, and you can start again. Down the rabbit hole you go. Scrolling down here, you might find the bibliography has been found. Of course, if you keep going back in time, because obviously you can't cite something that goes in the future, so if you follow the bibliography, you're going back in time. You will eventually find documents that are so old, nobody knows anything about them. They're probably OCR'd and therefore all better off. But certainly with recent articles, which is kind of the main workhorse of what most scientists have to care about, you have pretty much everything at your fingertips. One other thing that it can do, which I will briefly explain before stopping talking. There are some possibly quite interesting, not necessarily that useful, I think, for some biologists, but for others, very, very useful indeed. If you go through an article that I mentioned earlier, you might have some tables or some pictures or some graphs or those kinds of things. If you wanted to do some analysis on those in the page, it would otherwise be impossible to do, obviously, because the data's hidden away. If the article is well curated, if it's got enough information about it online that this can be done, Utopia will automatically turn, for example, a table into an actual table with values that then you could, if you wished, decide to graph, for example. And then you can bury, well, you can burrow down, I should say, into the data that's being shown on the document without ever even leaving the application. As well as that, there are some other things, the Harry Potter effect of being able to actually look at 3D structures in the paper, which, as a computer scientist, I think possibly not that brilliant, but enough biologists have said that that's actually really useful, even if all it is that when they're reading a paper, they just want to look at the other side of that molecule and they haven't got an image of the other side of that molecule. Yes, and so with the right kind of information available and with publishing getting better every year, the things that we can now do with what otherwise would have been static, dead PDFs are now helping scientists do their job more efficiently. And that's, I'd like to think, that's a success in terms of the reason I wanted to do research in the first place. And that's why I do what I do. So thank you very much. That's the end of the demo. I'm amazed that the internet lasted long enough for that demo to work, because it never does. But otherwise, if anyone's got any questions, feel free to ask.
Has anybody got any questions to the day? Or comments? Duncan.
Oh, sorry.
Plans for the future?
We'd like to get Utopia integrated into various publishing pipelines, kind of the reviewing stage. Because most of the time when you're reading a paper, a scientist wants to essentially vet the information in it. Either because they are reading it themselves and they want to see how it applies to the work they're doing, or it's because you're a reviewer and in the peer review process you're trying to work out whether or not something is useful. And this too, as far as we can tell, and certainly some of the publishers we're working with, I think it's got a lot of potential in terms of speeding up and cleaning up the reviewing process. So that's one direction we want to go. some of the stuff we're currently doing we can't really talk about due to various agreements and waiting for people to be able to launch some ideas. But,
is this part of becoming really, really rich?
Whereas obviously I would be lying if I said I didn't want to be really rich. Probably quite enjoy it. It's certainly not our goal. We'd like eventually to open source some of the things within Utopia, because some things like how the document structure is decided upon behind the scenes, that kind of thing could be really useful to everybody. So we're working towards getting that open source, which means kind of bypassing various partners, affiliations and things, because some people don't necessarily want us to open source it. So yeah, we want to make enough money to be able to continue doing it. But beyond that, I certainly have no interest in making lots of money.
Which publishers have been the most difficult to work with?
The larger ones. Not because they are not interested, but because of the politics involved in trying to get them to do anything. The largest publishing companies, if you want to work with them, you maybe have a conversation with one committee who have to take something to a different committee, and then a different committee. And then 2 years later, everyone's kind of forgotten what all the talk was about and you haven't done anything. Small publishers, on the other hand, and we've worked with quite a few now, they are keen, they are eager, and we can get some fantastic results. When we launched Utopia, we launched it with the Semantic Biochemical Journal. The publishing company Portland Press Limited, one of their journals is the BJ, the Biochemical Journal. They used Utopia to mark it up. So one of the documents that I just showed you, the one with the molecules and the rest of it, that's one of their curated articles. And for the last 3 years, all of the articles from that journal that have been published have had extra information embedded into it for people to use. So yes, smaller publishing companies, larger publishing companies, eventually.
What about outside of the biomedical domain?
Outside the biomedical domain, we've had a few bits of interest. We've had chemical, so biomedical and chemistry in my head are two separate things. The Royal Society of Chemistry pre-annotated, I think, every single one of their documents using Utopia documents. So if you open up any RCS, one of their documents, then you get access in the document to all of the supplementary materials and definitions that they would otherwise provide on their website. We've also integrated it with archive for physics papers. Essentially, it's possible to do this with any field at all. It's just a case of getting enough people interested and having enough hours in the day to write plugins for it. Because the core of Utopia is agnostic to the domain you're talking about. There's nothing in Utopia that's just bio-related. It's only the plugins that provide that extra functionality that's currently leaning towards biology.
Okay, time for one quick question because we have to finish in just a sec. Anybody else got anything they'd like to ask Dave? Comment?
Don't feel obliged.
No. Okay, well thanks Dave, that was a great talk as always.
Thank you.
And can we say thanks to Dave again? Thank you for coming.
Thank you.
Hope to see you again. Yes.